Hey guys, welcome to Biomix Hub. Today in this video, we will be discussing about the DNA damage response. So when a cell is dividing, it is undergoing various phases of the cell cycle. And during these phases, there are some of the checkpoints which are present. If there is some of the damage in the DNA, these checkpoints will not allow it to go from one phase of the cell cycle to the other phase. That means if there is some of the damage in the G1 phase of the cell cycle or something is not present which is required, then these checkpoints will not allow the cell to go into the next phase. So what happens when a DNA is damaged, how the cell is going to respond, we will see in this video. So in the last video, we have already discussed about the cell cycle regulation as well as a G1 S phase transition. Now, let's see how if the DNA is respond, how the cell is responding towards this. So when there is a double-stranded break, when there is stack of the replication, that means the replication is not taking place. And if there is replication, but the DNA has some of the mismatches, or when there is nucleotide damage, it activates some of the protein or some of the kinase like ATM as well as the ATR. This ATM is specifically activated when there is double-stranded break, and ATR is activated when there is single-stranded break. These are the kinase. This further activates some other kinases like CHK2 and CHK1. This ATM, it activates the CHK2 and ATR activates both CHK2 as well as CHK1. This further help in the repair mechanism if there is some of the minimal damage in the DNA. Otherwise, it will inhibit the CDC25. The work of CDC25 was that it was activating the CDK2, which was helping in the transition from the G1 to S to M phase in this manner. So if there is inhibition of the CDC25, there will be no activation of the CDK2, and that will take place. There will be blockage in the transition. When the DNA is getting repaired, then only it can enter into the next phase of the cell cycle. The next thing is that it activates the P53 protein. This P53 protein, it does two work. If there is little damage, it will activate P21, which will further repair the DNA. If there is more damage in the DNA, it will lead to apoptosis. So let us see how this is taking place. If there is some of the X-rays or due to some of the chemicals, there is a damage in the DNA, it activates the protein or kinase, that is the ATM and ATR. This further activates some of the kinase like CHK1 as well as CHK2. So how this work? What happens after the CHK1 and CHK2 is activated? There are two proteins. Like we have already discussed that there will be activation of P23. So how does P53, how this P53 is regulated? It is regulated by another protein which is known as MDM2. When the MDM2 binds with this P53, there is degradation by proteasome. That means this MDM2, it is a ubiquitination ligase. That means it will add the polyubiquitin into the P53, and there will be degradation. But when the CHK2 or the CHK3, CHK1 or CHK2, it will phosphorylate the P53. It cannot bind with the MDM2. The efficiency of binding of MDM2 with P53, it will be decreased. So this MDM2 is released now. So here the P53 is stable. There is no degradation of the P53. So what was the work of the P53? It was leading to apoptosis, or it was activating the P21. Okay, now this P53 which is present, it will help in the synthesis of the protein, that is P21. Now this P53 is working as a transcription factor, okay, which is helping for the generation or for the synthesis of the P21 gene. So here there is a P21 mRNA, and here we are having a P21 protein. So what is this P21? This P21, it is a CDK inhibitor protein. That means it is inhibiting the work of the CDK, that is cyclin dependent kinase. So here there was activation of the G1S CDK as well as S1 CDK. Well, it is the MPF as well as a cyclin, CDK as well as a cyclin. Now they are in the combined form, which is known as MPF. They are activated. That means this helps in further transition from the G1 to S phase or S phase to M phase. There is a transition, but when this P21 will come and bind in this region, there is inactivation of this G1S CDK or S CDK complex. So what happens during this time? there will be no further transition. That means where the cell is present, in which phase of the cell cycle, they will stop there. After the DNA has repaired, after the DNA is fine, if there is no mutation or if there is no damage, then only it will help in further transition. Till then, this P21, which is a CDK inhibitor, it will inhibit it. So let us see how it is doing. So here we can see that that is double strand break damage. So it is activating the ATM. ATM is activating the CHK2. The CHK2 is phosphorylating the P53, by which P53 is increasing its level. There is no degradation of this P53, and it is activating the P21, which is binding with the cyclin E CDK2, by which that is G1 phase arrest. So when the DNA is damaged in the G1 phase, such type of regulation is taking place. So that is another protein which is known as MYC. If there is excessive MYC production, when there is excessive MYC production, when there is some of the damage. Okay, so what this MYC does is by further transition, by further process, what it secretes the ARF. This ARF binds with the active MDM2 and make this MDM2 inactive. Now what happens? The P53 is stable now. It is not degraded by the help of MDM2 because this MDM2 is inactive now. So when this P53 is activated or when it is stable, it undergoes apoptosis or cell cycle arrest. We can see here when there is double-stranded break, there is formation of ATM. When there is single-stranded break or unreplicated DNA, that is synthesis of ATR, or these two proteins are activated. ATM activates CHK2, ATR activates CHK1. The CHK2 and CHK1, they inhibit CDC25A or CDC25C. When there is inhibition of the CDC25A, this blocks this activates the CHK2. Okay, when there is activation of the CDC25A, what it does it blocks the CDK2, which will arrest the G1 S phase transition. When there is activation of CDC25C, it will inhibit the CDK1. The CDK1 is inhibited and the arrest will be in the G2 phase. G2 phase arrest will be taking place. So here again we can see how it is done. When there is single stranded break, that is formation of ATR. Here there is inactive CHK1. After this, there is phosphorylation of the CHK1. When the CHK1 is phosphorylated, it phosphorylates CDC25. The CDC25, which now after the phosphorylation, the CDC25 is activated. The CDC25, what is this? The CDC25 is a phosphatase. It inactivates the CDK. Now the CDC25 is working as a adapter protein for 14-3 sigma. Okay, so when there is some of the ionizing radiation, when there is a single stranded break, it is due to ultraviolet radiation, but when there is a double stranded break, it is due to some of the ionizing radiation. When there is some ionizing radiation, there is formation of mRN complex. So that is activation of ATM. This ATM, it activates the CHK2 by phosphorylation. This CHK2 is phosphorylates the P53, which was unstable. After the phosphorylation, it is stable now. Then it further activates this P53 work as a transcription factor and it activates the P21 which activates the which inactivates the CDK. That is here we are having a CDK which was activated. Now it is inactivated when the P21 is binding in that, and that is a cell cycle arrest. So this was all about the DNA damage response during the cell cycle events. So in the next video we will discuss the M phase transition. Thank you.